Hello everyone, my name is Hunter, and I'm one of y'all's MS1 buddies. I thought it would be good to kind of give my own version / a supplemental version of the Lecture 8 we had, since I thought some of the topics our professor went over were kind of confusing. Here is me in the top right, my GroupMe profile picture with my amazing dog, Nikki. In the top left, we got some happy lungs here. Lastly, I want to mention that I actually do have something to disclose, which is that I used to have / still have a ribcage deformity known as a sunken chest, which messed a lot with my ability to breathe growing up. I was able to get a surgery during high school to have it fixed, and during that time I got really interested with lung physiology / medicine in general, which is why I'm here at BCM today. Anyways, I just wanted to add that as motivation for why I really care about this material, and let's get started. All right, so here are the 3 objectives for the presentation, literally just our professor's objectives, just rearranged. Essentially, what I will do is go through each of these in kind of my own way, and I'll be adding some supplemental information / a different way of rearranging the material that I thought would be helpful. Again, I just wanted to say that my lecture here shouldn't really replace our professors, but instead hopefully supplement it and explain a lot of concepts that I thought he kind of either assumed we had knowledge of, or I guess just hopes that we'll go over at a later time. Also, the description in this YouTube video will have timestamps for each of the objectives I go over, so if you want to just click on a specific objective, you can just go ahead and do that instead of having to watch the whole video. All right, so before we tackle any of the objectives, I think we should talk about the idea of respiratory volumes, which our professor kind of mentioned but didn't go super deep into really explaining it. It could be that we'll go over these later, but I really think this is something that everyone should know before going over literally like any other lung topic. So the main takeaway here is that if we plot the lung volume over time, we can get some different lung performance parameters depending on what action we have the individual do. So first we have this idea of tidal volume, which is basically involving the shallow breaths we expect an individual to do, and kind of like what y'all are probably doing since you're not doing anything too strenuous and you're probably just chilling. So then if we ask someone to breathe in as much as they can, they hit this upper limit at this respiratory capacity, and then if we ask them to breathe out as much as they can, out, we reach this limit here called the vital capacity, which is the total delta V anyone could input and output from their lungs if they ever tried their very hardest. So that's the vital capacity here. Then with this vital capacity, we can actually use this for different respiratory ailments in diagnosis. So for example, before I had my sunken chest surgery, my doctor actually gave me some spirometry tests to actually see just how bad my breathing was with my chest indent. My doctor said that my vital capacity was about 70-ish% of what my vital capacity should have been for my demographics. So I definitely, qualified for the surgery, and hopefully I'm at a better spot now since then. So the next thing we should talk about is the residual volume, which is essentially the leftover bit of volume that you can never really get rid of from your lungs despite how hard you try to breathe it out. So in other words, because the physiological exhaling action of your lungs is only so good with a biological system, you can never actually get a pure vacuum in your lungs once you try to exhale as hard as you can. There's going to be some air inherently left over, and that is called the residual volume. And lastly, after the residual volume, if you combine that with the expiratory reserve volume, you basically get this idea of the total amount that sits underneath the lower bound of your tidal volume. So from here to this delta V from functional reserve capacity, which is the combination of just everything below lower bound of your tidal volume, you get this delta V, which is, I don't actually know how to motivate it super well in an abstract sense, but I do know this is something that our professor talks a lot about in these lectures. And if you're ever wondering where the FRC or functional reserve capacity comes from, this is where it comes from, and we're going to see how it affects diseases of respiratory compliance later on in this lecture. So what I think would be useful in order to understand the pressure gradient airflow in the lungs would be to kind of get a rough estimate and oversimplified model of what's going on here with the interpleural pressure forces along with the expansion forces of the lung and the chest wall. And of course the diaphragm along with the chest wall. So we're just going to lump the chest wall and the diaphragm together. So let's say this is the chest wall plus diaphragm. And this is of course tied to the parietal pleura, as our professor had talked about. And then over here, you have the interpleural space, so the IPS. And this interpleural space lies between the visceral pleura that is on the outside of this lung. And so you have this interpleural space, and there is a pressure associated with the interpleural space. Let's call it the PIP. And there's also a pressure associated with the alveoli. I don't know if it's big A or little a, but let's just say it's big A. And then due to the difference between this PIP and the alveolar pressure, there is a I think it's a transmembrane pressure. And so the transmembrane pressure is the driving pressure gradient that changes the actual size of the lung itself, and the alveolar pressure is the driver of air going in and out of the lungs here. Now, the PIP is negative. It's very negative. It's like -5 compared to the atmosphere pressure. All of these like integers are just like in reference to the atmospheric pressure. So this -5 here, it's important that it's negative in the interpleural space. It's like just a property of the different like cells that excrete fluid into this interpleural space. There's like a feedback regulation that I'm not sure if we're ever going to learn it, but that's basically what I found through my Google searches. It just is a fact that this space is negative. Now, what's important here is that because it is negative, that basically acts as a transmission of force between these two areas. So I'm actually going to draw this on the other side here now. So if you have an outward pull here, that is going to increase the volume of the interpleural space, which is going to decrease the pressure of the interpleural space. And now with a decrease in the interpleural pressure, this transmembrane pressure is going to drive an expansion of the lung tissue to account for this. And what's going on here is basically this force is transmitted by this interpleural like negative pressure going on here. So because it is negative, the forces are basically transmitting inward of the walls, pulling both of the walls towards each other. So as this one becomes, this kind of outer wall gets pushed outward, it is going to have a opposite force that then pulls the lung outward. And so you would have this increase in the actual lung space. And then with the lungs being pulled outward in this kind of simplified model, you would then pull out the alveolar diameter then out this way as well, which is going to then pull air into this space. And then with a decrease in this chest wall diameter, it's going to result in an increase in the interpleural pressure. That is then going to push this pressure down into here. So it's going to squeeze on the lung space, which is then going to push the lungs back inward on itself to then go and squeeze this air it had just gotten out. And so hopefully this was a little bit of a better explanation of what's going on. But in case you don't understand what's going on, I'm going to have a GIF/video playing later on that shows an animation of everything I just described, and that's hopefully going to help illuminate some of the actual cause and effect dynamics of what's going on between the pressure and the volume changes in expiration and inspiration. So what happens if we don't have this negative interpleural pressure? And what you can actually kind of see in this GIF, which is a bit of an oversimplification of what's actually going on, but the whole idea is that the negative interpleural pressure is what allows for the lungs to actually stay inflated and to actually act as this transmission of force between elastic recoil of the outer chest wall going outwards to then communicate that to the actual lungs. So in a world where you don't have this negative pressure space in between the elastic forces of the lungs and the elastic forces of the chest wall, you basically allow for the lung to be able to collapse in on itself because the lung elasticity and its recoil wins out. So something that's important to note is that in our professor's model, he notes that as the regions around the lungs change, so in this interpleural space, you have different amounts of interpleural pressure. And the biggest component of this is probably going to be the fact that gravity is pushing down in different areas and pulling in others. So it's going to be compressing down here, which would result in a less negative interpleural pressure, whereas in this other area up here, we're going to be having an expansion due to gravity, which would cause a more negative interpleural pressure in this space. But for the general model of how we're going to approach the pressure changes driven by volume changes in the lungs, we're going to mostly just assume that these gravitational effects aren't super big in terms of this lecture at the very least. All right, so I was able to convert a YouTube animation of the inspiration versus expiration cycle, where they show the volume and pressure changes and exactly what causes changes flow into / out of the lungs, and that's shown through this respiratory cycle GIF. Now, and I think that's pretty helpful to understand the concepts. The idea is that in response to the outer wall / diaphragm, increasing the thoracic cavity or decreasing the thoracic cavity, you're going to result in a change in the interpleural pressure, which is going to drive the lung to have a size change, which is then going to either push or pull air in / out of the alveolar areas. And yeah, that's basically all that's going on, and I think this GIF is really helpful in showing these volume and pressure changes. So now on to objective 2. So we're going to summarize the factors that contribute to air resistance. Now, all of these basically stem from Poiseuille's law, which is our old friend from the cardiac lectures. So the resistance is equal to 8 times the viscosity times the length over pi and then r to the 4th. So there becomes 3 kind of logical chunks that stem right away from this equation. So the viscosity term, the length term, and the radius term. So with the viscosity, what he talked about here was the idea of decreasing the viscosity using the addition of helium to the mixture of gases the patient is breathing in. I believe he said this was a treatment for asthma. So if you are breathing in a mixture of gases that has a lot lighter or perhaps less viscous gassy properties, then I guess you are able to decrease the resistance that way. So that's way 1. So next is the length term, which the professor actually didn't really give an example about, but I guess the whole idea, as you can kind of see from this top area, is that the resistance that the gas is having to put up with is a function of the length of the tube the gas is going through. So if you decreased the length, you would actually result in an decrease of resistance. Now, what's interesting is that when you actually blow up the lungs, so you're going from a small lung boy to a big lung boy, you are actually increasing the distance that the air has to actually travel to get to the alveoli, which actually means that there's a bit of a negative feedback loop, because as the lungs expand with air, there is therefore increased resistance that the air going into the lungs has to travel, because there's an increased length they have to travel to actually get to the alveoli as the lung expands. So fun fact, professor didn't mention it, but I thought it was cool. Lastly, we're going to be talking about the radius. So here, as we all know from the cardiac lectures, this is where if you are messing with the radius, you're going to mess with the resistance by a factor of 4. So it's either going to be in, by doubling or halving, you're going to be influencing the resistance either by a factor of 1/16 or by 16. Now, the way this is manipulated is, of course, with our other friends, cholinergic and adrenergic receptors. So, of course, with the sympathetic nervous system and then the parasympathetic nervous system. So with the sympathetic nervous system, you can cause dilation, and this is done via beta 2 receptors. And then the parasympathetic, you cause constriction of the bronchioles, by the way, for both of these, by muscarinic receptors. So typically, I don't know if there's ever a case you'd really ever want to constrict an airway intentionally, but if you did, you can activate those muscarinic receptors, and then in the world where you want to dilate, you'd either inhibit the muscarinic receptors or you would activate the beta 2 receptors. Lastly, let's talk about this idea of airway tethering, which, to be honest, is very hand in hand with Poiseuille's law as written here. So the idea is that when you're at a small lung space, there are parts of the lung outside of the alveoli which will expand and contract with the expansion and contraction of the lungs, and these influence the actual path the air will take to get to the alveoli. So let's say that you have this lung. Here is, I guess, the midline of the body. So when you breathe in, you would cause an expansion out this way in this kind of tube that the air is flowing through. And not only is there an expansion this way, there's also these little sources of elasticity that pull the pathways in the air and just kind of all the parts of the lungs towards themselves, towards where they started from. So there's through the inherent elastic recoil of the lungs. So even though they are expanding and expanding, there is this inherent tethering force that pulls them back. And so what we actually have here, basically like an elongation and almost like a ellipsoid elongation in this model of what's going on. So instead of it being perfectly circular, it's more ellipsoid because you're kind of pulling it in these kind of two different directions. One, because of the normal expansion, and then one because of the tethering. And now because of this, you are overall increasing the cross-sectional area of the actual airway the air would go through. So this is the final cross-sectional area compared to the cross-sectional area you started with. And because you're increasing the cross-sectional area, it's basically analogous to an increase in the radius going on here. This Poiseuille's law is really only derived for if you're in a perfectly cylindrical tube, but in our more ellipsoid tube, it would be a similar effect where either way, an increase in the cross-sectional area would decrease the resistance. Therefore, as you increase the lung volume, i.e. you expand the lungs, you result in a decrease in resistance. So onto the last objective for this lecture. So we're going to define the pulmonary compliance and the two main factors that determine compliance. So here is our curve that's going to help us demonstrate exactly what is going on with compliance. So compliance, according to our first aid book for step 1, equates to the change in lung volume for a change in pressure. So delta V over delta P, which is exactly what you're looking at on this graph, i.e. the slope of the tangent line at each of these points, whether for inspiration or for expiration. The slope of the tangent line to the curves is going to equate the compliance. So EPIC Calc 1 application here. So increased compliance is going to mean an easier time filling the lungs, and additionally, compliance is also equal to 1 over the elasticity. And I believe the elasticity implies the ease at which the lung actually returns to previous shape. So if you have a very compliant lung, it's going to be really easily deformable and really easy to expand, but because of that, it's really hard to actually get it to contract back and recoil back to its previous small boy ideal state. So now there's two parts that make up the factor of the compliance. There's going to be the elastic recoil and then the surface tension. And I'm going to be talking more about the surface tension in terms of surfactant later. But in terms of now, we can actually use the surface tension, and in particular, the intermolecular forces associated with surface tension, the intermolecular forces of the hydrophilic portions of the things like water vapor and other things like that that are just kind of around the alveolar space, not necessarily like messing up the idea of ventilation, but just kind of there, and they're contributing to some forces that inhibit the want to expand the lungs during inspiration. So for example, let's use a smaller alveoli as the initial, and then you're going to try to increase this space. Now, what is basically the main effect of why this curve shown on the left has a weird shape during inspiration is that there is a point where the surface tension is actually causing a really difficult time for the alveoli to expand. So with the surface, there's a lot of hydrophilic interactions that are going on here that are displaced a bit by surfactant, which I'll get into in the next slide. But the whole idea is that there's increased surface tension in the world where these little molecules that are hydrophilic are way closer together, as opposed to the world where they're a lot more spread apart. It's way harder to have these intermolecular forces that dominate, wanting to keep this lung in the more collapsed space. So the idea is that you have the surface tension here that is playing a part, that is fighting against of trying to expand the lungs. But eventually there becomes a kind of inflection point here, somewhere around here, that results in there being a decrease in the surface tension forces, like the radius of the alveoli has expanded so much that the surface tension no longer plays a part. And so suddenly the slope and the compliance is able to rapidly increase compared to where it was over here. So compliance is way down over here, and then compliance is way up over here. And this is because you overcome the surface tension, and so it's able to very easily increase its volume for a change in pressure. But then eventually you get to another kind of petering off here once you get to the maximum volume that the lung could really take in easily. I don't know if this curve is actually corresponding to tidal volume or maybe vital capacity. Didn't really say. I think it's just more in theory here. So hypothetically, if this were the limit of tidal volume, then the person should be able to breathe in a little harder and actually increase his upper limit. But because we don't know, we're just going to assume that this is that they're not going to be able to expand. So once they get to this max volume, it's just downhill from here. So it's really down, down, down as the lungs then deflate and air is pushed out. But then there becomes a point right around here where suddenly the rate of decreasing becomes more and more negative. And that's because the compliance in the expiratory direction, so expiratory compliance, is actually increasing. And the reason for that is now the surface tension is working with the decrease in the size. So once the molecules get closer and closer together, eventually there becomes a critical point where they become so close that they want to increase their intermolecular forces towards each other, which is going to therefore decrease the actual radius of the alveoli, which makes it easier and easier to actually deflate fully once you get to this kind of critical mark here. So this is an epic, I guess, Gen Chem explanation of why this is all happening. But now let's actually get into how surfactant plays its role in the surface tension. So now let's talk about surfactant's role in the compliance. So compliance, is the ease of filling the lungs, and that is going to be increasing with increasing amounts of surfactant. So what is surfactant? That is basically, as the professor talked about it, it is lipophilic molecules made by the type 2 pneumocytes, and the idea is that they are going to disrupt the hydrophilic forces that are normally present in alveoli. So disrupt hydrophilia. Okay, and it does this not just by being lipophilic, but they're also amphipathic, i.e. The actual main component, the DPPC, actually messes up the hydrophilic interactions because it is amphipathic and it is actually a phospholipid. So fun fact, so it has, of course, the hydrophilic end and the hydrophobic end, and it's able to mess up the hydrophilic interactions that would normally be caused by water and other hydrophilic molecules that would be found in the alveoli. So what would be a world without surfactant? Let's look at that real quick. So the idea, if you're in a world without surfactant, you would have kind of the alveoli as a little ball crumpled in on itself. So very small, and because of that, it is really hard to actually get air to flow into here to actually expand. So it's actually really difficult to expand in a world without surfactant because of the increased intermolecular forces here due to the hydrophilia kind of causing increased ball-like energy going on in the alveoli. But with surfactant, sorry for my handwriting, by the way, I don't know if it's really annoying, but you are instead able to have a more rounder shape where it is less collapsed in on itself. So let's say this is the surfactant here. And what this does, it is better able to disrupt those intermolecular forces here. So there is less of a collapsing force going on here. Therefore, it is more likely to stay in this easier to inflate state. So it is easier to inflate because you are disrupting the intermolecular forces here. And so yeah, so just to recap, with surfactant, you cause a decrease in the intermolecular forces. So you decrease the collapsing force, which means it is easier to inflate to a big alveolar boy. So here is a really cool graph I found to be able to actually appreciate and understand what surfactant is actually doing in terms of the compliance here. So this is without surfactant here, and this is with surfactant. And even if you just look at the average compliance curves of both of these graphs, you can see an insane increase with the presence of surfactant. I think this is actually just an artificial one too, used in this study. So it's not even the real thing. It just goes to show that going from a world where you have no surfactant to then a world where you even have this artificial form, it's kind of insane just how much easier it is for the lungs to actually be able to expand. So for example, after 20 centimeters H2O of force that your lung is trying to do to increase the actual size of your lungs, you get a huge difference in the actual size that the lung is able to actually expand to with and without surfactant, because it is just way easier in this top curve to actually be able to expand the lungs, whereas in this bottom curve, you're trying so hard and applying so much pressure differential, and it's only around the 20 to 25 mark into the 30 mark that you're actually able to do any kind of work here. And this is such a small amount of actual volume work going on compared to the 2.5 times the volume load an individual alveoli can actually have in the top curve. So I don't know if you want to just kind of sit back and appreciate what's going on here, but it's really cool to really show just how much compliance can be changed by just the presence of this surfactant, and how kind of amazing it is that we have this in our lungs. So I'm just going to have to hope that you understood what our professor was saying in terms of the expanding forces and collapsing forces shown in this diagram from the slides. I do want to relate, though, that the FRC that he talks about here is the same FRC I had talked about earlier in my kind of intro into Objective 1. So this functional residual capacity, just everything that is below the kind of lower bound of the tidal volume here. So now we're going to actually talk about the clinical correlations and how they relate to the compliance and FRC that can be shown on the plots that our professor talked about. So the big two here are emphysema and fibrosis, and hopefully they can be easily explained through the concepts of compliance and FRC. So, just to reiterate, the FRC is this boy over here, as we just said, and that can actually be shown on the chest, lung and chest wall graph C here. So with emphysema, the idea is that you have an increased FRC, and then with fibrosis, you have a decreased FRC. I believe this part of the graph where these kind of dots are show the translation of FRC with each of these. And then in terms of compliance, it's FRC. And then in terms of compliance, these can actually be shown here, I believe, in the slope of the tangent line once more. So going to emphysema, you would have an increase in your compliance, and then fibrosis, you would have a decrease in your compliance. So what does it actually mean to have increased compliance and increased FRC? And let's talk in particular with the emphysema case. Now, I actually found some cool study where it compared the person with healthy lungs versus someone with COPD. I believe COPD and emphysema are interchangeable in this case, but I really hope it doesn't come back to bite me that emphysema and COPD have wildly different clinical presentations, and that it's totally learning it wrong and things like that. Hopefully it's as useful, this explanation of motivating why FRC and compliance are important things to care about in terms of lung function. So hopefully this becomes a good example in hindsight, but I guess we'll see. Now, the idea with compliance, i.e. a decrease in elasticity, is something you can actually see on this kind of right side of this diagram I have here. So with a decrease in the elasticity, you're actually increasing the size of the alveoli end at compared to the normal lungs that are much smaller in the normal size. And the effect of that is that with the decrease in the elastic recoil that would contract it, you are starting off at a higher baseline of the volume you are dealing with, but the total upper bound of the volume you can get to is constant, right? So it's not like you have bigger lungs here. You just really screw over how low you can actually get on the lower bound of air. And so because of that, as you can see here, you're elevating the actual amount of the functional residual capacity, which is a total bad thing, kind of bottom graph here, because as you can see, when the person is trying to exercise, they're having to do a lot more work in terms of breathing in and out to actually do the same delta V. And you can't even do the same delta V. The delta V just gets way smaller, so you have to do an increased amount of work to actually do the same amount of gas exchange, I should say. And so they have a much worse ability to actually expel carbon dioxide and to take in more oxygen. So it's probably way harder for this person to actually be able to, exercise well. And it's because the lower bound is at a much greater spot. So there's just less work you can get out of and less actual function you can get out of the work involved, because with trying to breathe in as much as you can, it doesn't really make as much of a difference because you're starting off at a much greater volume. So the actual total change you breathing in does is way diminished. And so I think that's a really cool clinical correlation of what it actually means to have something like emphysema, and hopefully that's communicated to y'all why this kind of concept is important. So now let's talk about the flip side of this for our last slide. So fibrosis with a decrease in compliance and a decrease in the functional residual capacity. So I wasn't able to have a really cool volume versus time graph, but for this one, I had some bar graphs that can helpfully go and illuminate some of the core concepts going on with emphysema versus fibrosis. So let's talk about the actual idea of fibrosis with elasticity first. So what's important to note from this graph is that the residual volume between the normal lung function and the fibrosis really doesn't look like it's changing on here. And what's important to note is that you still have the normal alveolar starting size. So it's not like you have a greater starting size like you would with emphysema, because there's this way bigger residual volume. We don't have that here in the case of fibrosis. So it's the same starting out in terms of the volume here, but it can be the fibrosis one, this can be the normal. What happened though is when you try to expand in the fibrosis, maybe there's a thicker wall here or something like that. So it is just way harder to actually do this expansion work here, whereas in the normal case, it is way easier to expand it here. So what you can see here is that even though the starting volumes are the same, as you can see on both the normal and the fibrosis graph, you're doing less delta V. So because of that, increased elasticity and recoil, much stiffer lung we're dealing with. So it's way harder to actually do the same amount of volume change compared to the amount of work we're putting in, the amount of pressure work. So with the same amount of diaphragmatic contraction and the same amount of chest wall expansion, it doesn't matter. You're not going to be able to increase the lung size as much as you would in a normal patient. And that's how that compares to fibrosis now. And yeah, so with emphysema, you're probably trying to do a lot more, work of the additional inspiratory muscles with the chest wall to actually do the same amount of work in terms of breathing. So you're probably having to exert a lot more energy. And typically the people associated with these patients have a lot more of a puffed out chest compared to the normal person, and they have a barrel lung kind of chest presentation. And that's because they're stuck at just a higher end respiratory point compared to the average person. So they're probably having to do a lot more work with their extra inspirational muscles to do the same amount of breathing work, which of course is probably very strained. So yeah, hopefully I touched up on all these concepts and help y'all motivate why I think a lot of these topics are important, hopefully with some different explanations of the material. Hope to see y'all in the next lecture. Please DM me any feedback or any issues you found in the PowerPoint. I can make some corrections. And yeah, good luck with studying. Hopefully this video wasn't too long. Hopefully it was helpful.